koutou kua huhui mai nei I tēnei rā. Ko Libby Liggins, aho. I'm the chair today and I'm really privileged to invite our first speakers. We've got Helen, Chris and Claire telling us about their important project.
Tēnā tātou kua huhui mai I rungi te kaupapa kotahi. Imhi ana ki te iwi kāinga ko Ngāti Whātua me ona hapu. Whai kite kia koutou katoa. So I just welcomed everybody and also acknowledged the iwi for this area and saying it's really good to see you all. I'm not familiar with all this sort of stuff. Sorry, I better introduce myself a bit. I'm Chris and I have been working together on and off for 8 years, I think it is, so I've grown in my knowledge of genomic science. But I'm a psychologist by training, and I've been working in policy and strategic direction for the National Te Whatu Research Program. And that's an exciting journey. So, but I'm here today because I'm going to talk about how we began, how this journey began, and the people that were involved in it, and some of our accomplishments and some ethics. So I just wanted to introduce you to Dame Neira Rangimaria Glabis. She's a rangatira for the local mana whenua, a local tribe, and also the Chief Advisor tikanga for Waipapa and Auckland hospitals. In the beginning, when I first came on board in 2013, their iwi had already begun to talk about storage of tissue. And then the next piece of information that they were discussing at iwi level was, I think it was biobanking, and then it was genetic research, and now it became genomics. And so Chris and I, Chris plucked up the courage, I went with him to see the dame and ask her if she would give us a name, because of course names carry with it certain histories, and so that's what that first slide talks to. She's our tikanga patron for the Rakeiora program. The other person that we approached was Paparangi, because we needed some clinical guidance, and we had several conversations with her, and she said to us that she doesn't totally think that genomics is the way forward, but she agreed to be our clinical patron. And I think that it's always good to have somebody who disagrees, because that keeps you sharp. So I just want to introduce you to the people that are part of our kaitaki group, bit by bit. So this is Irene Kiriama Royal. She's got a legal background, but also worked in genomics and what do you call it? In the environmental space. I say 30 years and they tell me 20. Some of you may know Professor Maui Hudson. His background when I first met him was around ethics, and then it grew into ethics and biobanking and ethics and genomics. So he sat on our group and sometimes joins now, doesn't he? Anyway, he helped establish our thinking. Similarly with Professor Wilcox, he's recently became a professor and is very excited about it. His background also is ethics and environmental genomics, but him and Chris and Stephen Robertson were secured funding to establish this group. What have I done? I was just gonna, it's coming up over here, but not over there. Thank you. Well, thank you for that.
It's okay on screen now.
Okay, thanks. Some of you may already know Andrew Spoyle. He's a statistician, been working in Stats New Zealand for years, and also was integral in the cancer, inherited cancer, stomach cancer. And then we've got Ben Tiaka. He's actually a carver, steeped in tikanga mātauranga Māori. But I think he's done other things because he's been talking about the treaty and the law and things like that, hasn't he? I must push the wrong button. And then there's Chris and Kimmy, Lynn Sadlier and Sarah. So each of them came, actually Chris is part of our kaitiaki group, and so is Kimmy. Lynn and Sarah, Lynn's a very experienced statistician, and Sarah is a clinical trials leader. They're both doctors in their chosen practice, and so they've been helpful when we started thinking about, when I have issues around clinical practice in a hospital setting, or if I don't fully understand what's being said in terms of stats, and I trained as a statistician, but it's been years since I've had to use it. So they helped us. And finally, we were very fortunate to meet these three people who, when we talked about, what we wanted in a database, they actually absorbed it, absorbed the tikanga and the mātauranga, and they went away and they thought about it, and they produced things that answered our questions. But they also spent a lot of time with, is this a star thing? Oh yeah, oh no. With Phil Wilcox, the top middle there, and Ben, growing it a bit further and developing their understanding. And this is Tuti Watson. She's actually the chair of our organisation, and I'm sorry she came last because she's very important. Should have put her first, but never mind. She won't mind. So as a group, what we did was establish some standard operating procedures, and we used all these documents. I'm not sure whether you're familiar with them, but if you are, you can contact Chris or myself for the references. So what came out of that, apart from, we had several standards. We had ethics, treaty, tikanga, and Māori engagement. But at the beginning, Ben Tiaka talked about the Treaty of Waitangi, and in that he makes quite clear how Māoris are to be treated in research projects, really. And I had some notes. Those are the things that he wrote about keeping Māori researchers safe in the, as research partners and things like. I'm sorry, I've just lost my notes. I'll leave it. He had other things to say, and it's just a small part of what was in our standard operating procedures. And we don't, there's quite a lot in that document, and I'm just going to talk to these three Māori ethics, and as you're reading it, I'm going to tell you a story about what that looks like in practice. So about 18 months ago, a Māori woman came and agreed to participate in a clinical trial. And so she was gifting not only her tissue, but her whakapapa. And when the person who was consenting her came to talk to her about it, and the person who was taking the blood from her came to take the blood, she put her head against the person's head. And that's a spiritual connection, whakapapa to whakapapa. And then So in that sense, we had, all the investigators had a responsibility to carry that with them wherever they went. And so it's a spirit, carry the spirit of the gift of the tissue wherever they went. And then at the end, which is te whakahoki or te taonga, returning the gift, they had to bring that gift back to the person in one shape or another. And sometimes I think we call it, what's it? Kawitonga. So what we bring back as researchers, maybe, is we have a hui, and at that hui we talk about the findings. And in the findings we might say, this is going to benefit Māori a certain way, and maybe not. But it's taking it, you know, we're not just taking it, we're giving it back. Sometimes it's given back as a hui. I've given back as a book. I never authored it, of course, it was my colleague, and I'm not sure where that book went. But it can be anything. One thing I don't think is good is money. I don't think that's about returning the spirit. But sometimes people give money if it's in their heart to give. So I guess the message for me to you all is that the success of our progress was about us, was about the people in the group, our willingness to work together, our respect for each other, our consideration for each other. And I have to say that I think some of us might love each other as well. Thank you. Claire, your turn.
Ngā mihi nui, Helen, thank you for that wonderful introduction. Kia ora koutou, I'm Claire Rai. So as Helen hinted, I was part of the NESI team who are also here today, who were involved in more of the platform development. I think in the essence of time, I'll just touch on a couple of points. And Helen really gave us a lovely introduction into the starting, into the beginnings. And one of the really key, I guess, sets of principles that was developed with Helen, with Ben Tiaika, with most of the group earlier recognised, was this development of the tikanga attributes. And this was really, I guess, pillars for us as a development team to understand what was needed and to evolve our thinking in order to create a safe home for this data and for the analysis, and really ensuring that we respected sort of the spirit of the gift in the aspects of where we held that taonga. I'll actually skip over the platform aspects just in the essence of time, but I'm very happy to talk through that both from I guess a researcher point of view and interactions, and also from the kaitiaki point of view. And we really tested this with both the primary care and tertiary care cohorts as they came through. Very briefly, from a researcher point of view, one of the key aspects around access control was not having access to the data directly, not being able to download and manipulate, but being able to, in a controlled environment, carry out analysis. And so we had a sort of a standard GA4GH visa passport mechanism, and this is just a screenshot of seeing in our test system actually of the sort of the type of visa and information that I'm allowed to have access to, whether that's full genome or whether it's actually just a region of the genome. And then in terms of actually starting a job, and Ben Halliday's had plenty of practice at doing this and testing our system for us and finding lots of bugs when we were actually running it, in terms of this is how you'd set up to do the analysis. So this is a tertiary care setting, so we've got both the tumour sample sequence and the normal to compare it to, and then you get sort of a log as it's running, and then output files that you can then further analyse. And I'll hand over now to Chris to talk through the sort of logistics, I guess, and a summary of the tertiary care setting. Thank you.
Kia ora. So, as a non-Māori researcher, you approach this sort of project, which we were really, I guess, tasked with building a nationally scalable prototype for whole genome data and health data for anyone across New Zealand. It was a te tiriti partnership, and we approached it initially in a very Pākehā way, thinking we'll build the infrastructure and we'll consult widely, etc. But our collaboration took us on a most wonderful journey where, as you saw, there was a starting point going right back to the core principles, which are Māori principles, which enrich the project so strongly. From these, Claire described tikanga attributes, and Claire and Nick and Jun and several others at Nessie then hardwired these tikanga attributes into a nationally scalable computational system using best of world visas and passports and so on. And this was then applied to two ends of the clinical research spectrum. At one end with Stephen Robertson, Huti Watson, Phil Wilcox and others in Ngāti Porou Oranga, whole genome sequencing of individuals and whānau across New Zealand in a primary care or general practice setting. At the other end, which Helen and I co-led as another form of te tiriti partnership, this was focused on cancer patients in Auckland in a tertiary care setting. And this is a slide. This is a slide just showing part of the patient information sheet. And I thought it was a nice way to illustrate what patients expect as they come into the project. They have their normal tissue and their cancer, in some patients up to 6 different metastatic tumours, whole genome sequenced and sequenced with a targeted panel, and alongside this they have kindly given permission for their health information and information held about the health by the government to be linked and consolidated in a research platform. After the linkage and consolidation, the data can be stored federated around New Zealand wherever people are comfortable, but the analysis is done within a walled garden where the data never leaves the walled garden, never goes out to someone's computer, certainly never goes out overseas. This is just one screenshot with an example of the type of analysis we see. In columns are individual patients' tumours, and in rows are genes with mutations. And sorry, you're not going to be able to read this from a distance, but the top gene with the most mutations is TP53, the most commonly mutated gene in cancer, so there's no surprise that's up there. You'll see some patients' tumours had huge numbers of mutations, all of these little blocks as you go down, where other patients had just a single mutation. The way we did this has allowed us to really understand well how whole genome sequencing of normal tissue and tumour, how targeted panel sequencing, this is just sequencing genes that are really important for cancer, work with different types of patients in different clinical scenarios. But all of this in the context of this incredible Rakeiora infrastructure with the hardwired tikanga attributes which we are so lucky to have in there. So just want to finish off with the concept that Helen began with, that this is a te tiriti partnership to generate a nationally scalable database for whole genome sequence data and a range of clinical data together. You could imagine this growing in the future, and we hope it can. The tikanga attributes were reduced to practice by an incredible team in Nessie and hardwired into the infrastructure, which allows walled garden analysis, so the data never leaves, but federated storage. And as you can see, there are teams here from a whole range of different regions as well as a whole range of different fields. There are people who do the DNA sequencing in the lab. There are data scientists who design the infrastructure. There are clinicians who have referred the patients in and are returning the values to the patients. And of course there is Ngāti Porou Oranga strongly represented. Thank you very much. Kia ora.
now for questions, because I think people would likely want to have that put over with you. So, any questions for the team? Well, feel free to follow up with them at the lunchtime. Thank you, everyone. Where is your one, Jim?
Mine's 12 labels.
It's in this
Oh,
one, isn't it?
oh, yeah.
Yeah. Okay, our next speaker was to be Prasad. I believe he can't make it, so we have Jun speaking on behalf of him and the team.
Cool, thank you. Kia ora, my name's Jun. I'm from Digital Research Innovation and AI team at Waipapa Taumata. Prasad, Babarenda, Gamage, my co-presenter couldn't make it today, so I will be speaking from the perspective of both the Auckland Bioengineering Institute as well as our team with our very recent collaboration work. So I'll start with a bit of background of the 12 Labors project. It's built on Physium project that's been going on for decades, which with the goal of building a generic digital twin of human that integrates existing knowledge of molecular cellular biology, anatomy, physiology, and physics. And the 12 Labors project picks up on that and builds on top of it for building personalized workflows and building a platform that enables patients to have personalized copy, personalized digital twin, so that clinicians can run like repeatable tests and find out what treatments may be best and also identify any serious health risks. Here are 4 exemplar projects that are being used to test the platform and also make sure to demonstrate the benefits and values. That includes biomarkers for pulmonary hypertension, rehabilitation of upper limb disorders, and monitoring of uterine health, and automated reporting of breast cancer. And each of these projects, they are leveraging on the development of each of these groups' work that's been going on for 10, 20 years prior. And all of these, at the end, they are producing clinical reports, and each of them have established clinical collaborations and specific applications. And in the end, the goal was to have these projects proceed to clinical trials and ultimately clinical translation and possibilities of commercialization. These are not all the things that's happening in ABI. ABI has 300 plus researchers with hundreds of workflows. So in the future, the platform, the goal would be to support all of these different work that's going on. Here's a diagram showing a little bit of link between different workflows and different studies. The 12 Labors team has adopted ISA standard investigation study assay, and each assay represents at a workflow level. So the first assay is showing a process of generating personalized lung model, and the output of that gets fed into the next workflow, next assay, where you use the personalized lung model to report underperfused regions, and you get the outcome of clinical report at the end. And that falls into the study of developing the workflow under the investigation of, in this example, estimating vascular remodeling in pulmonary hypertension. And that can follow with something like validation study. And here are some challenges that the platform was trying to solve. So these are existing challenges that include like loss of siloed infrastructure. It's difficult to know what other groups are working on. And with the processes of running the workflows, most of it was manual in the past, and it is difficult to scale. And as for conducting the studies, there are limited services available for rapid development cycle, and also it was very difficult to get access to clinician time as well as access to data. So the aim of the platform was to develop a platform that supports and accelerates the clinical translation of virtual human twin workflows. And here are some three key objectives: provide common infrastructure using fair tools and services, standardize and automate workflow data management, and make the execution of the workflows reproducible, repeatable, and also streamline development and evaluation of these custom workflows in both research and clinical environments. So the 12 Labors team has created a platform called DigitalTWINS AI platform, which stands for Digital Translational Workflow for Integrating Systems AI platform. And I will give a little bit of overview. So on the left there, you can see different kinds of data types that goes into running an investigation. So studies, models, measurements, tools, and workflows. And the platform has data ingress into harmonized database, and it becomes browsable through data catalog. So you can see what kind of data is there. And adjacent to it is a knowledge base that might not be directly related to the investigation, but is fed into workflow inputs. And all of that goes into workflow execution using interactive portal, as well as various dashboards and things like that. So the end users here shows clinicians, researchers, and even patients for something like patient dashboards. So some of the workflows might be running in the background, but some might have interactive elements, which I'll give an example soon. Under the hood, there's a Spark data set structure being used for FAIR data, and ISA standard, as I mentioned earlier, as well as the team has adopted NFDI for Health, which is German national infrastructure, and the team's building on the clinical metadata, and also building the workflows using common workflow language, but it's not limited to it. So you could potentially use different workflow languages as well as different tool chains. One part of the service is FAIR-DOM-seek, which allows you to add descriptions to the data so that across the tool chains, as the end users, you would be able to get more context around the data that you are dealing with. And as for the workflow management, the 12 Labors has adopted Apache Airflow for running and monitoring the common workflow language workflows so that you can see the progress and what went wrong and results and things like that. So all of these workflows, they can be triggered from the portal under the context of the assays and studies, and you can view the results, but there are things that allow you to do interactive evaluation and things like that so that you have access to existing applications like Jupyter, but there are also tailored applications that can be added to the workflows, so, such as this one where partnering clinician Anthony Doyle has co-developed. So this allows Anthony to report on the position of tumors, and this is a manual process, manual step in the workflow, so he would go into the web interface and do that. So this, they are working towards a clinical pilot to be deployed on Te Whatu, and this is the part that our team has been collaborating on, and this is based on 900 patient study design, and the goal is to compare the accuracy against ground truth segmentation for automated reporting and manual reporting from clinical records. So this is where our team comes in. With our collaboration started at Q4 last year, and our main goal was to help with the robustness and scalability of the platform as we are going into the deployment into Te Whatuora. And we wanted to also be able to work together using good development practices and also help out with some of the identity integration challenges. Existing platform already used good practices in terms of containerizing all the services and using Docker Compose and using GitHub for all the changes, and they had 4-5 developers contributing to it. And what we've done on top is we try to Ansible-ize it, so we added as code approach to the deployment itself, so with a little bit of Terraform, and we added things like GitHub Actions, so that using dev test branches for respective deployments, and if you make the right commits, it goes into the dev and test environment directly, and you can see the progress of the deployment and things like that. And we also helped with the backups and recovery, and we are introducing Kubernetes so that it is more future-proofing as opposed to Docker Compose. But we are starting with a single VM deployment solution. So we've looked at something called K3S, which is minified version of Kubernetes, and it's our first time trying deploying Kubernetes with that, but it has worked out really well. Another aspect that we are helping out is with the platform logging and monitoring. So we're using technologies like Grafana, Loki, Alloy. Our teams actually all from formerly Nessie, and we have used this technology stack a lot in the previous HPC and all the services that we've been developing. So this screenshot shows uptime across all the different containers and services. And you can do things like keyword search within all the error logs and message logs, so that if you know certain phrases belong to certain commonly occurring error, you can set up alerts and things like that. So this collaboration is built on our engagement with ABI over, I'd say 2-3 years, but it actually goes longer. From Nessie days, we've been sharing the same office building, and our team's approach has always been looking for that long-term alignment. And our dev team, two of our team, Matt and Carbon's involved, and the 12 labors developers, Chin and Lincoln, so 4 of them are contributing to the code at the same time and working very closely together. And we are constantly looking to lift the capability and share the knowledge both ways, and is always relationship-based. So going back to the DigitalTWINS AI platform, a bit of a long-term vision of them. So currently they're hosted in ABI as well as Nectar, and with the pilot, the goal is to have the pilot in Te Whatuora in a secure environment, and help with the supporting clinical translation and possibly commercialization. And even longer term vision is for the 12 labors DigitalTWINS AI platform to be usable beyond the university and nationwide, or even internationally. And MedTech IQ, it's a possibility there. And Prasad team has also been in talks with lots of international partners, and Sant Pau in Barcelona, one of the oldest hospitals in Europe, has also agreed they will onboard the pilot soon, and UC San Diego after that. Also, based on these developments, we just heard like a few days ago, 12 labors were included in a Horizon Europe infrastructure grant, which used the platform's blueprint. So this is really good news. And here are all the networks from the work. And I would like to part with thanking everyone involved. That's me. Thank you.
Thank you, Jim. Super neat stuff. I've got a question toward Jim. Chris.
John, it's amazing. Can I ask a question about the last slide where you talked about international linkages? A lot of people are generating these sort of things internationally. Is there a standard emerging that's going to allow interoperability?
One of the slides that I actually removed from here because I didn't know too well, but Prasad had was about the FHIR standard, FHIR, and I think that comes into play here. And from that, all the standards that they adopted, they are trying to adopt all of the international standards. So, Claire, do you have better answer?
So it seems this pilot you spoke about with DigitalTWINS is around the automated breast screening. So I'm guessing that in that case, and probably all the times you roll it out, there's going to be a bit of training of the clinicians and a little bit of feedback from them on improvements. So I guess, because it goes back to Chris's question, it's sort of some parts are going to be completely sort of generic and be able to be interoperable, but there's going to be some bespoke tweaking in certain circumstances. Is that something that, how much work, I guess, do you foresee with that? And is that part of what you are expecting with the pilot, that kind of, modification as a result of the clinicians' input?
Yep, I think key part of that is as they are developing these workflows, they have partnering clinicians like Anthony Doyle, who's been working with the processes even before the context of 12 labors. So I think they are constantly developing these processes. And as for maybe deeper answers, I may have to pass on to Prasad to actually, understand the full plan.
They're running both protocols, from what I understand, in parallel. So they're going to run the current protocol alongside the new protocol.
Just to catch any
Mm.
gaps.
Yeah, learning and there's lots of moving parts into PhD and with different teams. So lots of opportunities.
That's really neat.
I have another bite. I'm just wondering whether there is a real opportunity here. We were incredibly lucky to have Genomics Aotearoa support Rakeiora and a number of other projects, and the educational side of this is going to be critical, isn't it, to really make this work, the clinical capability development side and scientists and data science in particular understanding the clinical needs. I wonder where we're going to fund that, and I wonder whether that's the sort of thing.
I just wanted to Chloe, really like a Chloe training opportunity.
Yes.
There. Yeah. Have a conversation around that. I think there's a whole lot of work to be done in that space. I mean, also, you and Chris, but Emma is talking today around the visual cell modeling stuff, and linking all that in with large language models. Training around that would be fantastic as well. So, yes, I think there would be a pretty substantial training opportunity there.
Yeah, absolutely. I just rolled back so that there's actually education dashboards which Prasad has been trying out. So I'm sure he'd be really keen to seek the opportunity there.
Fantastic. Thank you, John.
Thank you.
So we're going to have a little mini break here. I'm happy for people to have a bit of discussion time in that if they want, or nip out with a drink or whatever you need. We'll be starting again on the dot at 11:45. It was a bit late for the last session, sorry. So next speaker, please come on up and close the talk. 3 minutes.